you're currently working with these or you are thinking about implementing iPSC-derived organoids into your research programs, today, by the end of the presentation, you will have taken away, hopefully, some valuable information for these workflows, including methods for developing organoids from iPSCs, how to develop scalable 3D workflows, whether you're sorting suspension-grown spheroids or organoids, if you're interested in clonal organoid development, and monitoring differentiation of iPSCs into organoids. And then finally, how to create custom 96 well plates with single organoids of interest for downstream assays and assessment. Induced pluripotent stem cells have sparked widespread enthusiasm for the development of new models for human disease. And this is partly because they've eliminated the ethical concerns of previously used embryonic stem cells, but also because they provide potential to study disease and patient-specific aberrations. These disease-specific iPSCs could either be derived from patients with a known disease or mutation, or by taking normal, healthy donor iPSCs, performing some kind of gene editing that's specific for the disease of interest, and then creating cell lines for those oncogenic or disease mutations. With the advancements in iPSC technology as well as organoid technology, these iPSC cultures can be directed via growth factors that mimic the in vivo niche to self-organize and differentiate into organoids that mimic both the cellular and the structural complexities of tissues. These models provide greater physiologic relevance compared to 2D in vitro models or animal models for therapeutic and clinical testing, including understanding developmental disorders as well as disease pathologies. They're very useful in the drug discovery pipelines as well as in clinical research and also in toxicology testing. Now, despite the potential of these models to improve that in vitro to human translation in the therapeutic pipeline, it's widely known that as the complexity of your model system increases, there are inherent challenges preventing them from being employed in high throughput applications. And that starts with scalability, both from a cost and ease of use perspective. Many of these workflows, particularly when we're talking about differentiating iPSCs, are incredibly manual, which not only reduces your efficiency and increases your labor cost, but also introduces variability that can impact your reproducibility. And that's both well-to-well within assay plates and across multiple assay plates or biological samples. And it's because of these challenges that the use of organoids as an in vitro model are generally limited to low throughput applications. So I'll pause here for poll question 1.
We'll just give 20 to 30 seconds for that poll. You can answer the question on your screen. Okay, thank you. Go ahead, Allysa.
I'm not able to advance, Lisa. There we go. Okay, so in today's webinar, much of what I'll be focusing on are neural organoid differentiation workflows. However, this animation, this schematic, represents what a lot of 3D iPSC differentiation workflows look like in terms of having similar, very highly technical manual procedures. In this workflow for generating neural organoids, we start with our 2D iPSCs. They are seeded into 96 well plates with thousands of cells per well for aggregation and embroid body formation. After embroid body formation, these are pipetted manually into another well plate format to combine those organoids per well for neural induction. After neural induction, each of those individual organoids gets seeded into an ECM droplet. And then once those droplets have polymerized, those organoids are moved into the 6 well plates for expansion and differentiation. So you can see here that these steps, which involve not only manual pipetting and replating, but a lot of time and labor, has the potential to induce a lot of variability into the process. This can not only impact the success of your protocol, but ultimately can impact the organoid formation and the utility of your model downstream. Today, I'm going to show you how we have collapsed this multi-step protocol onto a single consumable, reducing both your hands-on time and increasing your efficiency. And in addition to simplifying the culture methods for these organoids, our automated workflow produces multiple custom assay plates with single organoids in every well, whereas this manual approach has an output of only 96 organoids in bulk suspension culture. And importantly, we are able to do this using 65% less media, ECM, and consumables. So today, I'm going to introduce you to 2 technologies that we've leveraged to develop scalable, automated 3D workflows to demonstrate how you can perform these methods in your own lab. I'm very excited to introduce to the first time a technology that's new to our 3D portfolio. We've added the Cero 3D in partnership with Omni Life Science to offer a solution for high yield suspension cultivation of 3D models. The Cero 3D is an approachable mini benchtop bioreactor that maintains temperature CO2. It can perform online pH monitoring and has the opportunity to run 4 independent experiments in the instrument. Now, using the Cero in our lab, we've grown a broad spectrum of 3D cultures, including embroid bodies formed with a SMOD inhibitor neural induction media. We've co-cultured 2 different lines of edited iPSCs into spheroids. We've also grown adult stem cell-derived organoids in bulk. So here you're looking at mouse hepatic organoids that you can see there in suspension. And then, of course, we've tested many different cancer cell lines for your cancer spheroid models. Now, these bulk propagated cultures can be used for population-level screening and analysis. But what if you're interested in investigating this heterogeneous population on a single organoid or spheroid level? And that's where the CellRaft technology really shines. Using the CellRaft technology, we can culture spatially segregated organoids, either seeded from those preformed cultures or grown directly on our CellRaft array. And then we can isolate those single organoids into 96 well plates for downstream use. For the remaining time of the webinar, I'll be showing you how we use the CellRaft technology and how you can leverage it to generate those custom assay plates at scale. The CellRaft array provides a flask-like culture environment for culturing cells while also providing that spatial segregation. We can then use our software and instrumentation tools of the CellRaft technology to identify our cells or organoids of interest. And then once we find them, we're able to transfer them into 96 well collection plates without any manual pipetting, fluidics, or enzymatic dissociation. And while this technology was originally developed for 2D workflows, we found that these features were uniquely suited to fill this unmet instrumentation gap in automating organoid workflows. So to tell you a little bit more detail about the technology, the core of it is the CellRaft array. This is an innovative cell culture plate. Cells are seeded onto the CellRaft array, much like you would seed many other tissue culture consumables. And importantly, they share that same contiguous media volume like they do in bulk culture. However, within that reservoir of the array, we fabricate an elastomeric microwell array that provides the spatial segregation of all of those cells sharing that contiguous media. At the bottom of those microwells, we fabricate a microscale polystyrene growth surface that we call the CellRaft. And importantly, these CellRafts are releasable from those microwells to be retrieved for downstream use. So just to orient you a little further here, you can see in white, we have that elastomeric microwell. At the bottom of the microwell sits that magnetic polystyrene CellRaft. And as I mentioned before, the really important feature of this consumable is that while those cells are spatially segregated, they're sharing that contiguous media volume, which allows them to exchange growth factors and have the crosstalk like they do in bulk culture. Using the CellRaft array, we're able to culture organoids with a little bit of a different approach that I like to call outside the dome. The workflow for generating these is really very straightforward, and it starts with our 3D CellRaft array. This is our largest CellRaft format. Each of the microwells and CellRafts are 500 by 500 microns, so we're able to grow much larger organoids. When we're performing our iPSC organoid workflows, we start with our iPSCs, enzymatically dissociate those either into single cell suspension or fragment suspension, seed them onto the array in an extracellular matrix suspension, and then after polymerization of that ECM, we use our CellRaft air system to perform 2D serial imaging to monitor organoid development over time. Here on the right, you can see we have a single iPSC as it starts to form that cerebral organoid over time. But what's really so impressive about the CellRaft array is the scale that we're able to perform these differentiation workflows. Each of the 500 micron CellRaft arrays has over 2,100 available positions, which means we're growing hundreds of organoids spatially segregated on a single consumable. Here you're looking at a stitched together image of the entire array. Those green circles are highlighting CellRafts that have an individual organoid. Importantly, we're able to image each of these CellRafts reliably over time because of that microwell array. This not only allows us to characterize our organoids ahead of isolation, but importantly for these iPSC workflows, it allows us to monitor differentiation over time, so we're able to collect data and really predict our outcomes downstream. So to show you what this looks like for our neural organoid differentiation protocols, here we're using a commercially available kit for developing cerebral organoids. You can see the arrow pointing to a single iPSC as it undergoes embryonic body formation, neural induction, and then expansion out to day 10. We've also performed these workflows using a choroid plexus differentiation kit. Similarly, through the first 10 days, you can see that fragment of cells in this case as it starts to form an organoid, and then through choroid plexus differentiation out to day 14. So just to reiterate, I'm showing you this series for a single CellRaft and a single organoid. But we are able to capture these time course data records for hundreds of organoids growing on that CellRaft array. So in addition to monitoring differentiation over time, one thing that is widely accepted in organoid cultures is population heterogeneity. And in the organoid space, this is actually a good thing and representative of population heterogeneity. In addition to monitoring that differentiation over time, the CellRaft technology really allows researchers the ability to investigate these heterogeneous populations on a single organoid basis. And that's not really possible in your standard culture methodologies where you have these organoids grown in bulk. So to provide a clear visual representation of what population heterogeneity looks like, we've performed these workflows by co-culturing iPSCs with 2 different fluorescent markers. So here in this image, you can see we have red organoids, we have green organoids, and then we have some that are actually expressing both fluorochromes. The beauty of the CellRaft technology and the CellRaft array is that you're able to parse out each of these individual phenotypes and look at them on an individual raft basis. So looking at these images, you can easily imagine the opportunity that this allows you to not only understand the heterogeneity, but also tailor your downstream applications based on the specific phenotypes that you're looking for. And the best part is we don't make you do this alone. So you're probably thinking that hundreds of organoids and all of those images sounds like a lot of data. Our CellRaft cytometry software has algorithms to help you find your cells and organoids of interest. That includes analysis across multiple time points. We can look for single cells or clusters of cells. We can also look for organoids downstream based on their diameter, their shape, or their dimensionality, and even fluorescence markers. So to give you an idea of how we are using the software experimentally in performing these cerebral and choreplexus workflows where we're generating clonal organoids, you can see here on our initial time point, we've assigned an algorithm to identify single cells. Then 10 days later, we're able to identify organoids based on size or fluorescence. And then using this population set feature, we can take those populations that we've built and overlay them to find our clonal organoids of interest. So you can see here that not only are we able to identify our cell rafts that have our organoids of interest, but we're actually able to use the software to quantitate the metrics of our differentiation process. So here you can see we have some data on 3 arrays for each of those organoid types. In the dark blue bar, we have our total single organoids, so an average of roughly 225. And for each of these workflows, we're able to achieve 50 or more clonally derived organoids in that workflow. This is a clonally derived organoid forming efficiency of more than 17%. And while that number may sound actually small, in talking with researchers and scientists who are developing these protocols, many of them have told me they've never seen a clonally derived cerebral organoid before. So I'd like to highlight the importance of the CellRaft array to be able to kind of unlock the potential for developing these clonal organoids. Now, once we have used these software tools to help us find the organoids that meet our criteria, we can easily map them to collection plates for isolation. We can also use the CellRaft array in a different workflow to segregate and sort our bulk produced 3D populations. So here you're looking at the samples that were taken from our co-cultured iPSC spheroids grown using the Cero. And again, seeding those on the CellRaft array, you can see here on the left-hand side with our field of view, we're able to identify rafts that have red spheroids, green spheroids, and those co-cultured red and green combined spheroids. And then once we have created this list of rafts, we're able to then dispense or sort these populations into our 96-well plates for further use. And that's where the CellRaft air system comes in to automate what would normally be a very manual process of transferring individual organoids into the plate. So here on the left-hand side, you can see we have our CellRaft array. On the right-hand side, we have our collection plate. We would insert our magnetic wand to the right of the light source. And this automated video is going to walk through the process of what that CellRaft isolation looks like. So here on our CellRaft array, you can see we've identified a raft with our cells or organoids of interest. Mounted on the objective below the array is a release needle that releases that CellRaft from the microwell. And then because those CellRafts are doped with magnetic nanoparticles, they're picked up by that magnetic wand and transferred into the collection plate without any fluidics or dissociation involved. So the cells or organoids remain attached to that CellRaft, and they're very gently transferred into the 96 well plate. Now, as much as I like the animation to sort of orient you to the process, I also like to show what this looks like in real time. So here on the left, you're looking at a hepatic organoid grown on those 500 micron rafts. So this organoid is maybe roughly 300 microns in diameter. As I advance the video, you'll see the focal plane shift as that release needle releases the CellRaft from the floor of the microwell. And then the magnetic wand, which is that bright spot, dips into the array to pick up that organoid. After the software has confirmed that the organoid has been removed, it is then transferred into its location on the collection plate. And this process is repeated for each of the map CellRafts that you've identified. To produce these custom assay plates of your phenotypically selected organoids in every well. So now that we've been able to not only grow these segregated organoids, but more importantly, for our downstream assays, develop these custom plates, the opportunities for interrogating these and using them for your downstream use is limitless. But I wanted to give you an example of how we've been using this technology for downstream assays. So here we're taking a toxicity screening approach using those single cerebral organoids. In this particular experiment, we wanted to address a biological question, which is, can we parse out toxicity sensitivity between diseased and normal iPSCs? So for this, we've used a healthy normal iPSC line and an iPSC line that was derived from a Parkinson's patient with this LRRK2 mutation. These LRRK2 mutations have been previously shown to increase the sensitivity of the downstream neurons or organoids, both in vivo and also in vitro, to different neurotoxicants. So here we're using two different neurotoxicants. One is an insecticide, rotenone, and the other is an anti-cancer drug that's commonly used. The schematic of our protocol for developing these assays is here. So you can see we're either starting with those normal or Parkinson's iPSCs. On day 0, we're seeding them onto the CellRaft array in our embryonic body formation media. On day 5 and day 7, we're performing full media changes on the array very quickly for neural induction and expansion. And then on day 10, we are taking all of our imaging data, we're identifying CellRafts that have our organoids of interest, and we're using the CellRaft air system to isolate them into the collection plates, into maturation media. Those organoids continue to mature off of the array in the collection plates out to, in this case, day 43, when we then do our toxicity dosing on those single organoids. So now you can see here, we're linking this particular CellRaft to its downstream mature cerebral organoid. So on day 10, we perform those software metrics to identify the CellRafts. You can see here, these organoids continue to grow and develop and mature off of the array. So here out to day 37, you can see not only is this organoid growing in size, but we're also getting these advanced neural networks that are growing off of the main body of the organoid, indicating that we are, achieving that mature phenotype. And what's more impressive than having single organoids in every well is that by selecting those organoids based on size and morphology at day 10, even after 30 days of maturation in the collection plate where we're just performing media changes, you can see here that we have well-to-well consistency for our assay plate to perform this toxicity screen. So here you're just looking at replicate wells, and while the morphology does represent the heterogeneity of the population, we have roughly the same size organoids, and they all have these mature neural outgrowths from the main organoid body. So then on day 43, we treat those single organoids with a 5-point dose curve of our toxicants. So for rotenone, we used a dosing range from 1 nanomolar to 10 micromolar, and for doxorubicin, 10 nanomolar to 100 micromolar. And then after 72 hours of exposure to these toxicants, we used 3D cell titer glow to measure cell viability for our downstream assay. Here you can see we have our dose curves for both of those toxicants. In the blue lines, you're looking at our normal iPSC-derived organoids, and in red, we have those Parkinson's organoids. And you can see here that in both of these dose curves, we get this right shift of the normal organoids, indicating that we have increased sensitivity or a cell viability difference in our diseased organoids. So here for rotenone, our EC50s for the normal were 0.1 micromolar, whereas they were 0.034 micromolar for the Parkinson's. And then similarly for doxorubicin, we had EC50s of 14.1 and 7.4 micromolar. So in both cases, the EC50 values for those normal iPSC-derived organoids was more than 2-fold higher. In addition to being able to look at the sensitivity based on the organoids, I'd like to also point out too that across these 5 replicates that you're seeing for these doses, we have very small error bars, indicating that we had well-to-well consistency within our assay. So in this example, I've demonstrated the ability to generate these assay plates for a toxicity screening application, but really the application space for these is quite limitless. So in the neuro space, that could look like developmental and degenerative disorders, including Alzheimer's and Parkinson's, but these could also be used for cancer and infectious disease research, as well as our drug development pipelines. So in the beginning of this webinar, we reviewed these 3 key challenges that limit the utility and the accessibility of these very complex biological in vitro models for high throughput therapeutic and clinical applications. And I hope today that I've been able to demonstrate for you that the Cero 3D provides a solution for scalable 3D suspension culture, including spheroids, organoids, as well as iPSC differentiation protocols. And then using the CellRaft array and the CellRaft air system, we're able to simplify very manual workflows and ultimately create these reproducible assay plates for our downstream applications. And with these instruments combined, the scale at which we're able to do this is really very large, right? Whether we're starting with our bulk produced suspension cultures and sorting them on the air system, or you're growing and differentiating your iPSC organoids on the CellRaft array, we can do this at a much higher throughput than you can do in your lab with the manual traditional methods of iPSC differentiation. With that, I'll pause for poll question 2.
I'll just give another 10 or 15 seconds here. Okay, it looks like the answers are slowing down a little. Go ahead and end the poll. Okay. Thank you, Allysa.
And with that, I guess this concludes this portion of our webinar. We'll open the floor for questions. I'd like to invite everybody to come to our website, cellmicrosystems.com. You can find a lot more information about each of these technologies, as well as request a demo. Or if you'd like to discuss your particular 3D models or your applications, please feel free to reach out to me directly at alyssa.stern@cellmicrosystems.com.
Thank you, Dr. Stern, for that comprehensive and insightful presentation. We'll now move on to the Q&A session, and I see we've already received several interesting questions from our attendees. The first one I can answer, is it possible to get a video or slides? And we will be sending out the recording a couple of days after the webinar. So you will get an email with that recording. The next question I have is, what is the maximum size that organoids can grow before forming a necrotic core in your automated system?
That's a great question, and I think that's a little bit model dependent. You know, our adult stem cell derived organoids that have you know, a hollow lumen and are more epithelial in nature, we've grown those on the array up to 1 millimeter in diameter before isolation, and we can still continue to grow those off of the array. In terms of our iPSC organoids on the array, we're usually isolating them somewhere around that 500 micron diameter before we isolate them into the 96 well plates, because for the majority of those protocols, the differentiation process and the longevity of those cultures is much longer than the adult stem cell derived cultures. So I think that's a little bit model dependent. But I hope that answers your question.
How do you tag them to find heterogeneity?
The question was, how do we tag them?
Yes.
So, you know, we have done some co-culture organoid differentiation protocols to sort of very clearly visually represent those heterogeneous populations. In terms of you know, untagged, we can sort these organoids on the array based on their morphology. You can do kind of artificial growth rates if you've captured the images of those organoids over time. So if you're interested in looking at, you know, a certain size cluster of cells or single cells at an early time point, and then you want to sort that population based on a size somewhere midway in your protocol to make sure that you know, what you're isolating is actually you know, across a protocol of development more similar. And that's also possible. So fluorescence is not necessarily required. All of our algorithms are built on bright field analysis, with fluorescence being an added feature. So that sorting can happen either by morphology or by those fluorescent markers.
Will organoid dome get damaged while transferring them to another plate?
So I think the question might be, do the organoids themselves get damaged? So we're culturing these in a much more dilute extracellular matrix. So you know, traditionally when you're growing organoids in a dome, that has formed a very tight polymerization. The way that we're culturing these on the array is in a much lower concentration of matrigel, so it still provides enough support for that 3D growth, but we're not trying to push those organoids through a solid dome. And even with our most what I would call sensitive organoid types, being those organoids that are you know, an epithelial-like layer with a hollow lumen, we see that those are transferred into the collection plate fully intact. If we continue to grow those off of the array, we do recommend that you spike in ECM into that collection plate so they do have that 3D support to continue growing. But in terms of the isolation process, we don't see any detriment or degradation of the organoid itself because of the gentle nature of that transfer, that CellRaft transfer.
Okay, next question is, how does the organoid size affect the outcome of the assay? And what is the optimum size for each organoid to start induction or excluding it from further assay?
So I think that is also a model dependent question. You know, these iPSC organoids that have a much longer maturation after isolation, I don't think that it's as imperative that you select a specific size rather than just limit your size range so that you have that assay consistency downstream based on selecting for a smaller range of sizes. However, we've done some toxicity screening with our hepatic organoids, which have you know, a shorter longevity in culture. And what we've found is that, you know, selecting organoids in that 300 to 500 micron range versus smaller or much larger, the outcome of those organoids for the toxicity screen seems to be more robust. So I think that could be a little bit model dependent as well.
Okay. What is the footprint of the air system? Does it sit in the BSC?
That's a great question. It doesn't fit in most BSCs. I have seen some BSCs that have a little more, have a larger depth. But we actually have a custom laminar flow hood that we sell as an option to add on to the air system. So otherwise it's bench top, takes about 3 feet of bench space. And then of course has a computer with it, but whether you're putting it in a larger BSC or you know, adding on our laminar flow hood option, there are options for putting it inside a sterile environment.
Okay, great, thank you. Is this platform applicable to different organoid protocols, such as the vascularized organoids and microglia containing organoids?
We have not done vascular organoids yet, but I will say that my experience with developing the workflows for all of these different types of organoids is that the robustness of the workflow is what makes the successful organoid, not necessarily the array itself, right? The array is a culture environment, but is not depicting the outcome of those organoids as much as your optimization of your media and your growth factors for whatever organoids that you're making.
Okay. How about downstream applications? How are the organoids detached from the rafts for RNA extraction or any other application?
Yeah, that's a great question. So for our our RNA or transcriptomic endpoints. We've found that the rafts are they're really not inhibiting any downstream applications. So for our collaborators who have done a lot of transcriptomics with single organoids, they simply just either isolate into their lysis buffers or isolate into media and then pull the media off and add their lysis buffers without any interference of that raft with the downstream process. We even have one collaborator who has used some automated downstream processing for their transcriptomics without any problems of the CellRaft at all.
Okay. For iPSC-derived organoids, do you typically need to add more matrigel over the course of your culture with the media changes?
Sometimes, yes. So for the 10-day cerebral organoid workflow that I showed, over the course of that 10 days, we find that we've optimized the ECM concentration to be robust enough to allow for those media changes without needing to add matrigel back. However, if you wanted to do those differentiation protocols and leave the organoids on the array longer, so say you're doing some sort of population heterogeneity assessment and you want to actually use the array more like a cell-based assay, you can absolutely add matrigel at any point during the experiment with those media changes without any issues. We do just like to provide our expertise in helping our users use the right concentration of matrigel, because there is some boundaries in terms of being able to have enough matrigel to support growth, but also not have too much matrigel that would inhibit that isolation of the cell rafts and organoids.
Okay. Have you used this method for kidney organoids?
I just started working with kidney organoids, so I'm really excited about a suspension workflow that starts in the Cero to develop EBs and the 3D precursors for kidney differentiation. That workflow is in progress. We have done a more two-dimensional kidney organoid workflow on the array, which provided really great monitoring of differentiation. But that approach is a little more of a what I would call like a 2D, 3D hybrid rather than a isolatable 3D kidney organoid. So that's something that we're currently working on, and I'm really excited about that Cero air system combination for kidney organoids.
Okay, another question is, how about the imaging of these cells? Have you tried the immunostaining and then imaging?
Yes, absolutely. You can immunostain directly on the array. So we have done that for a lot of our 2D workflows, but we've also immunostained those the more epithelial-like hollow lumen organoids. We can Z-stack them. Of course, without confocal imaging, you get a little bit of variability once you get high up in your Z-stacks. But 100% you can grow native organoids and then do some sort of immunolabeling to identify, so to speak, organoids of interest based on that fluorescence inclusion.
Okay. That's all the time we have for questions today. Thank you, everybody. If your question wasn't answered, we will follow up with you afterwards. And as we conclude today's webinar, I'd like to extend our heartfelt thanks to Dr. Stern for sharing her expertise, and to all of you for your active participation. We would appreciate it if you could respond to a brief 2-question survey after the webinar is ended, and a recording of this session along with additional resources will be sent via email and available on our website in a couple of days. Thank you, everyone.